Man has always lived among a vast array of species, but it was only about 30 years ago that he discovered the code that diversity was based on, the genetic code. In today's program, we are going to find out about genetics, DNA, cloning, and genetic manipulation, genetics and animals and plant reproduction, human genetics. The genetic code is universal and is found in all living cells, in plant cells, as well as in animal cells. It is contained in the deoxyribonucleic acid molecules called simply DNA molecules. There are DNA molecules in the nucleus of every single cell of all living organisms, whether it's a bird, a flower, or you and me. DNA molecules vary from one species to another, but they're always made up of the same bases. It's as if we could write, using only a few letters, a host of different words containing all information on species and individuals. What does this new science, genetics, know about DNA? All living beings are made up of single cells. In its nucleus, each of these cells, whether animal or vegetable, contains not only all information defining the species, but also all information defining the characteristics of each individual of a same species. The name scientists have given to all this information is genetic inheritance. Genetic inheritance is contained in the nucleus of cells. It appears as a large, elongated molecule resembling a spiral ladder. This is the deoxyribonucleic acid, or simply DNA. Each rung of the ladder consists of minute units called nucleotides, or bases. In all the cells of all living beings, there are only 4 types of nucleotides, those containing adenine, thymine, cytosine, or guanine. The entire dictionary of life is written in those 4 letters or bases. Through repetition in different sequences, these 4 bases give birth to the whole spectrum of living things. Every cell in the body of an individual organism contains identical DNA. Thus, every one of the 60 billion cells in the body of an adult human being contains identical DNA in its nucleus. On these DNA molecules appear specific areas, genes, which we could compare to words. It is estimated today that there are 50,000 different genes which are responsible for both our human and individual characteristics, whether we are dark-eyed or dark-haired, diminutive or athletic. The various genes that replicate in our 60 billion cells are not all activated at the same moment. At given moments, some genes are activated to form new cells for our heart, liver, brain, and so on. So together, the 50,000 genes make possible the formation of all the tissues of the human body. In addition to the 4 types of nucleotides, or letters, and to the 50,000 genes, or words, DNA has even larger subunits. These are called chromosomes, and we could compare them to whole sentences. In humans, DNA is carried on 23 homologous chromosomes. The whole human chromosomal system could be compared to a factory consisting of several buildings and thousands of workers, the genes. When a human being is conceived, 23 chromosomes are inherited from the mother egg and 23 others from the father sperm. A new cell containing 46 chromosomes is formed. From this single cell will be formed all the cells of the adult human, each one containing 46 identical chromosomes, with one exception. The sexual cells, the sperm and eggs, only contain 23 chromosomes. When they fuse, they form a new being, combining the genetic codes of the father and the mother. Because of this incredible microscopic scaffolding, with so many millions of possible combinations, the scientists agree that with the exception of identical twins, there is only one chance in 10 billion that two people have an identical genetic inheritance. However, today it is possible to create genetically identical beings. This is done by a process which geneticists call cloning. The technique involves first taking the nucleus of a cell from the body of a living being, usually from the intestine or the skin. As we know, the nucleus contains all genetic inheritance information. The nucleus is then introduced into a female sexual cell, a non-fertilized egg, whose nucleus was previously removed. The genetic inheritance coded in the grafted nucleus comes to life, and the process of life has begun. The egg cell begins to divide normally. The new being can then be implanted in a uterus where it can continue to develop. Cloning, therefore, makes it possible to create an individual genetically identical to the one whose DNA nucleus is used. Cloning has been performed successfully on several species of animals. The first human clone was created in 1978 by an American doctor, Dr. Shettles. The clone lived 7 days until it was ready to be implanted in the uterus of a surrogate mother. But Dr. Shettles killed the clone and put an end to the experiment. The production of remarkable individuals in series makes some people dream, disturbs others. While cloning can create beings who are perfectly identical, genetic engineering can create new ones. Today, the genes of all living things, insects, fish, birds, or mammals, can be engineered so as to give us new transgenic species, such as feathered mice or giant rats. The DNA molecule, carrier of the genetic inheritance of all species, can be cut, spliced, and edited, much like the shots of a film. Genetic engineering of this kind requires the use of restriction enzymes, which select and cut DNA molecules in two. A gene can thus be extracted from a DNA molecule and grafted to another DNA molecule previously spliced by the same enzyme. We thus create a transgenic species, that is to say, a new species from the combination of the genetic inheritance of existing species. Genetic manipulation can also be applied to bacteria, and even to more complex beings, like mice and men. Cloning, genetic manipulation, devices to identify or create genes. Genetic engineering has the means to revolutionize life. Today we have both the knowledge and the means to alter the genetic inheritance of humans and other species. But what represents an exciting challenge to some is clearly disturbing to others. The 20th century has shown us that we cannot always rely on scientific instinct to avoid tragedy. True, we're not yet overrun with supermen, colonies of identical perfect babies, and Nobel Prize-winning clones. But frankly, the exact role of 80% of the human DNA molecule remains unknown. Vigilance is necessary, but it must not make us forget that genetics provides us with the means to improve the productivity and quality of animals and plants. Since man has deciphered the genetic code and been engineering genes, he has undertaken the remodeling of nature. Today, we make anti-freeze strawberries, tobacco plants which produce their own insecticides, potatoes immune to disease, by introducing genes and bacteria into the cellular structure of plants, genetic engineering is revolutionizing agriculture. Until just a few decades ago, crossbreeding was the only way to improve the quality of plants. By making the best specimens of a similar species or a different species reproduce together, we were able to emphasize certain desired qualities. But the process is slow. Crossbreed, sort, crossbreed again from one generation to the next until satisfactory stock or potential is achieved. Nowadays, the genes of a plant cell can be altered to produce a mature plant. In a matter of weeks, in vitro reproduction from an engineered plant cell could yield several thousand identical plants capable of transmitting their new features to their descendants. Nevertheless, this genetic revolution demands serious thought. For example, to eliminate the use of pesticides by growing plants that kill harmful insects is, to be sure, beneficial for the environment and economically profitable. On the other hand, what will happen over the long term to plants that have been genetically engineered? Will they maintain their properties after several generations? Or will they mutate in some unforeseen way? From another point of view, when plant forms are developed that are more profitable economically, they cause the disappearance of varieties which we might badly need. The old species could prove more resistant to new diseases or more adaptable to climatic change. The vegetable kingdom lends itself well to genetics, the animal kingdom less so. With animals, researchers lack precision when they attempt to place a new gene on the designated site of the DNA molecule. The first genetic experiments performed on animals were growth-related. Giant frogs and mice were obtained by transplanting growth genes. The foreign gene, transplanted somewhat randomly on the DNA molecule, produced unpredictable results. 4 times out of 5, the engineered egg dies, and among the embryos that do grow, most develop in undesirable form. The success rate is therefore very low. Currently, these transgenic animals have health problems and a shorter lifespan than the control animals. As in the plant world, experiments on animals have been many and varied. Researchers are attempting to produce cows and hogs whose fats would be polyunsaturated, that is, without harmful effects on the hearts and arteries of consumers. Attempts are also being made to implant in cows a gene which would enable the animal cells to produce the proteins cows need. Such cows would no longer have to ingest huge quantities of plants in order to find the proteins required. With each improvement in the technique of placing genes at the right location on DNA molecules, obstacles fall away. Man has at his disposal the means to force the hand of nature, but we will have to wait a good number of years before knowing what impact this will have on our way of life. Long before genetics, mutations were part of the normal process of evolution. But since man has been able to cut and splice genes from one species to another, from one individual to another, he has the power of choice. What he does with plant and animal life, he can also do with human life. And this raises complex ethical and legal issues that our society must first address. For the time being, the sorcerer's apprentices are playing God, ideas rushing through their heads. Who knows, for instance, how the association of in vitro fertilization with DNA recombination would turn out? This child has 3 mothers: his genetic mother, his surrogate mother, and his adoptive mother. The egg was taken from Suzanne, the sperm from John. Barbara carried the fetus, and Diane adopted the baby. There are thousands of test tube babies living all around the world. In vitro conception is modern medicine's answer to insurmountable fertility problems. This solution, however, raises problems of a moral and legal nature that frequently neither the partners involved nor the courts are able to deal with. But the innovative potential of genetic engineering may soon create ethical and legal challenges that are far greater. Today, in vitro fertilization techniques can be coupled with genetic engineering. One or several additional cells can be added to the fertilized cell. Such genetic manipulation may be able to prevent congenital malformation and eradicate hereditary disease. But it also opens the door to altering the human species. There are biotechnological firms ready to undertake this kind of genetic engineering. However, a number of obstacles still stand in the way. The human genetic code is complex, and we are far from knowing all the possible variations among the genes. A genetic flaw might successfully be corrected in an embryo, but in doing so, a pathology could be created that might only become apparent in adulthood. Furthermore, geneticists have not yet found a way to place a corrective gene directly on the desired location on the DNA molecule. At present, it seems that experiments of this type are restricted to animal cells. The procedure is often a matter of trial and error, not to say chance. Hence, out of 100 genetic mutations on animals, 90 produce undesired results, 9 useless hybrids, and, if all goes well, 1 produces the hoped-for results. But one day it will be possible to introduce into the human cell new genetic traits which will be transmitted down to descendants. Who then will decide the evolution of mankind? A number of geneticists have chosen to conduct their research in the field of predictive medicine. Almost 80% of physical and mental handicaps in newborns are caused by congenital malformation and hereditary disease. Early genetic detection of such handicaps may, in some cases, make it possible to skirt disaster. This is also true of phenylketonuria, a disease caused by a genetic defect which results in severe mental deficiency in the child. If that disease can be detected at birth through genetic analysis, all that has to be done is to submit the child to a very strict diet until the end of adolescence. As an adult, the person can then enjoy a normal life and even some of the delights of a varied diet. This being said, even if we are able to detect them more and more easily, we still do not know how to treat most handicaps that are transmitted genetically. The prenatal genetic diagnosis to detect possible diseases or predispositions itself also raises a major ethical dilemma. Who will decide a pregnancy is to be interrupted because an unborn child is discovered to be genetically predisposed to alcoholism or diabetes? Genetic diagnosis, in turn, calls forth the specter of eugenics, which is the improvement of the human race by the elimination of all people who are defect carriers. With eugenics, or the creation of supermen, genetics may soon have the means to control the evolution of man. When genetics will have reached the point where we are able to control evolution, it may be too late to ask questions. Now is the time to do so. This concerns us all.